University in the UK, in London, actually. And first of all, to say thank you very much to Holly for enabling me to come along and talk to you about groundwater and microplastics and a project that we're just starting up. So I mean, I've sort of divided this talk into three sections, really. First of all, I'm going to say a little bit about groundwater and what it's actually, you know, what it is, what it does, and so on. I'm going to talk a little bit about threats to groundwater and microplastics, of course, and then a little bit finally about the project that we're just starting now. So just a reminder, I suspect, for most people that groundwater is part of the water cycle. Everyone's very familiar, of course, with the idea of water evaporating from the ocean, sort of moving to the land, falling as precipitation, running off via rivers and so on as well, infiltrating into the ground. But just to point out that there's another section, if you like, of the water cycle, and that's the aquifers, underground aquifers, where water is held in rock formations, basically. Lots of different rock formations. I've just highlighted three main ones, at least three main ones in the UK at any rate. Karstic, where you get these rather lovely underground rivers and streams. Porous aquifers, for example, alluvial aquifers, and then sort of big fractured aquifers. But they're all aquifers and they're all water-bearing rocks. And basically, groundwater is, because we can't see it, at least not easily, it's very invisible to most people. And actually, this is very widely recognized. And in fact, this year, the UN Water Day was dedicated to groundwater, called Making the Invisible Visible. They are highlighting this year the importance of groundwater, what it provides, you know, what can go wrong with it, and so on. And following on from that, if anyone hasn't read it, by the way, and anyone's interested, at the bottom of the screen, there's the UN report that came out in connection with this World Water Day. So lots and lots of ecosystem services provided by groundwater. There's provisioning services. Of course, groundwater is an incredibly important resource for humans in terms of drinking water and so on. Regulatory services as well. Groundwater ecosystems remediate contaminated pollution, basically. And then there's various cultural services and so on. And also groundwater is very much part of the Sustainable Development Goals developed by the UN. All of the tasks in 6, 12 and 13, so clean water and climate change and so on, relates to groundwater. Funnily enough, groundwater is very rarely mentioned in the SDGs, but it sort of underpins an awful lot of them. And as I mentioned earlier, groundwater is really important in terms of humans. In West Africa, for example, I pulled these just off the UN report. Groundwater accounts for about 28% of total freshwater withdrawals. In Nigeria, about 70-odd% of the rural population and 45% of the urban population rely on groundwater point sources for drinking water. But it's very variable, of course. It really depends on the rock formations. In Cape Verde, groundwater isn't used at all. There's a complete reliance, or more or less complete reliance, on desalination, for example. So groundwater is, as a resource, is extraordinarily important, and I think this has really driven the increasing interest that there is in this. But just to mention, and this is where my particular interest comes in, groundwaters, or aquifers more to the point, are home to groundwater ecosystems. In other words, organisms live there, and this is quite an extraordinary environment, really. Aquifers are really challenging. Obviously, there's no light because it's underground. Often oxygen is quite limited. Food is also very limited. By the time infiltration has occurred through the soil, for example, most of the organic matter that's useful has been filtered out, if you like. So food's very, very limited. But nevertheless, these aquifers contain ecosystems, microbial biofilms, of course, and then microfauna, maybe rotifers and protozoa and so on, and also actually macroscopic invertebrates who are highly specialized. And the bottom right of the slide that's there, there's one of them. This one's from the UK. It's a little amphipod. They not suffer, but they undergo morphological convergence an awful lot. So look very similar. They're all colorless, of course. No point having pigment, really. They don't have eyes. No point because it's dark. And they often have very low metabolic rates because there's limited food. And the food web as a whole, the ecosystem as a whole, mediates by geochemical processes like nitrogen recycling and also remediates contaminants, pollutants. And of course, it's the microbial biofilm that actually has those reactions, but the microbial biofilm is then controlled to a certain extent by the rest of the food web through grazing and bioturbation and so on. So just to give a shout out, if you like, for groundwater ecosystems. So we have this very interesting resource and ecosystem. And of course, as you might expect, there are plenty of threats to groundwater. I've just listed some of the three big ones here. Groundwaters suffer, if you like, from long-term storage depletion, usually because of intense abstraction for not just for drinking water, but also irrigation, of course, as well, and industrial purposes. They suffer from salination, going salty, and what that means, of course, is that their use as a drinking water resource is quite limited when that happens. And this can occur in different sorts of ways. You can get flooding by seawater in low-lying areas. More commonly, you get the abstraction that I was talking about causes seawater intrusion, which is quite a problem. And then irrigation itself can cause salination and, if you like, destroy the resource. And then last, but certainly not least, groundwater pollution, which of course decreases suitability for drinking water. And we're very familiar in a way with nitrate pollution, for example, of groundwater, but the pollution I want to focus on for the rest of the talk really is groundwater pollution by microplastics. And this is, microplastics, of course, is an incredibly important and current topic. So far, though, it's largely been focused on marine environments and also, to a certain extent, freshwater rivers. So the map at the top of this slide, you can't see the details, but you don't need to. What you really need to see is the dark red areas, or the different coloured red tones, if you like, because the darker the red is, the more mismanaged plastic waste there is in the environment. And Stefan Kraus, for example, and his team have been doing quite a lot of work on that. And you might just be able to pick out one or two numbers, if you like, dotted around there. And at the time I used this graph, these were the only records of microplastics in groundwater. So you can see there's about 7 or 8 records there in the literature and reports and such of quantities of microplastics in groundwater. I'm sure there are a few more now, but there's hardly any. This is a sort of global assessment, if you like. So we actually don't know hardly anything at all about microplastics in groundwater, despite the fact that this is a really, really important resource. So what we do know, though, is that they end up in groundwater animals. So the bottom panel here with the sort of greeny bits, this was some preliminary data we collected on the amphipods I was talking about earlier, and lo and behold, these groundwater animals have microplastics inside them. So, and going back to the sort of connectivity idea, the fact that the aquifers, although they're invisible, are part of the water cycle, there's a lot of ways in which microplastics can end up in groundwater. So this is a bit of a busy slide, but what it's trying to do is to point out the ways in which you can get microplastics into groundwater from the surface. So we've got surface runoff into rivers, for example, and exchanging potentially with hyporheic zones and then groundwaters. We've got infiltration from, for example, agricultural areas into the soil, which of course in the end connect up with groundwaters. That all takes quite a while, I suspect, although we don't really know just yet. But there are, if you like, macropores or fast track ways of getting microplastics into groundwater, for example, through earthworm burrows, through managed aquifer recharge, of course, is one super way of getting microplastics very quickly into groundwater. So there's all different routes that how you can get microplastics into groundwater, and there's absolutely no doubt, really, that they're there. And potentially they're a problem, although we don't know at the moment because we don't really have the information. They're very slow often to arrive in groundwater. It takes a while for particles to infiltrate through the soil and into groundwater. But once they're there, potentially they can remain there for a very long time, because residence times in groundwater can be very long. So what we might be seeing at the moment with those very few numbers that I gave you earlier on the graph, we might be seeing, if you like, the tip of the iceberg, just the first advanced guard of microplastics making their way into groundwater, and the peak of microplastic pollution may occur decades into the future, a little bit like nitrates, really, I think. So once they're there, they can stay there for a long time. So we might be looking at legacy effects for hundreds of years, potentially. And there's increasing evidence of their impact on human health. Main ways microplastics get into humans, of course, is via the food chain and via drinking water. And we know that drinking water, a large proportion globally of drinking water is obtained from groundwater. So that is potentially quite a large pathway. We know that, that's a bit wrong, that third bullet point, actually. I mean, we know that they have impacts on surface freshwater organisms and food webs. So they can cause gut blockages in invertebrates, reduce fecundity. I mean, this is all published stuff now. Reduce survival, reduce growth rates. And we are beginning to assume that a similar thing is the case in groundwater, although we don't know, and that's something the project hopes to find out. They, as well as actually having a direct effect on organisms via their sort of physical occurrence, they can also interact with chemical contaminants, which sorb onto the surfaces, or perhaps are integral to the microplastics themselves. And they have a sort of Trojan horse effect, if you like, so they can import, the microplastics import these chemical contaminants more readily into living organisms. And once there, then they can have an effect, if you like, on the organisms. So all of this, if you like, is very much supposed. We don't actually know anything about it, which is, of course, where the project comes in. And this is what I want to talk about a little bit. So this is a project called Microplastics in Groundwater Ecosystems. Plastics, very proud of that. And I'm the PI on it. It's a 4-year project, and I'm working directly with some colleagues at Robertson and also colleagues at Birmingham, led by Stefan Kraus. Now, let me see, you should be able to, sort of big pause while I work out how to get to the, can I do that? Yes, that's the way to do it. Okay. So just very quickly then, the objectives of plastics are, well, there are 4 objectives. I really want to focus on the first one, which is sort of fact finding, if you like, determine the worldwide magnitude and establish the first global baseline of microplastic contamination in groundwater ecosystems. And this is really important because, as I said earlier, there's only 7 or 8 records of microplastics in groundwaters, and we don't know how it varies with geographic location, you know, and so on. The other objectives are more to do with pathways of uptake, microplastic uptake into food webs, how the properties of the microplastic affect the food web, and what happens if you change some of the environmental constraints, for example, carbon and nitrate and things like that. How does that alter the way in which they're uptaken? We also want to find out how they change the functioning and resilience of food webs. And finally, a sort of the end product, or one of the end products, I should say, is developing a global model to predict trends of groundwater microplastic pollution into the future under different scenarios, which is quite exciting in my view. So, but as I say, I want to try and focus on the first objective, which is actually finding out what's there across the globe. And this is where we're reaching out to potential collaborators globally. We've developed a standardized protocol for boreholes, springs, underground rivers, windows, if you like, into the aquifer. And what we're doing is people who've agreed to help us, we send the standardized protocol, obviously, but also sampling equipment, sieving materials, vials for storing the mesh and so on, to the collaborators. The collaborators then collect the groundwater from wherever they want to, so boreholes or springs or whatever. And once that's collected, we'll send a prepaid label, and then the samples are couriered back to us. And there's different ways you can collect these samples. Say let's just focus on boreholes, for example. Of course, they can be collected with a mechanized pump. This is on the left-hand side, there's a picture of a Grundfos groundwater pump. So you can do that, of course, with a generator or whatever. But also, depending on what's more convenient, really, you could use a hand-operated pump like this one here. That photo of hand-operated pump there was sent by a collaborator in Vietnam, or was it Thailand? I can't remember. Anyway, somewhere tropical, as you can see just from the background. And these are quite common in the area that this researcher works in. And, you know, it's a very simple way of collecting water, groundwater, and then it has to be sieved and sent to us. Also, we have a data sheet that's to be filled in, and it contains things like what's the land use, you know, how big's the borehole, and these sorts of things. Once we receive the parcel back, we can then look at what the composition of the microplastic is, what shape it is, for example, following the Plastic Rivers Protocol that Holly was highly involved in, of course. We can differentiate between primary and secondary microplastics. We can characterize the polymers and so on. So there's a lot we can then do with the samples. But of course, the main thing is we have to persuade people to help us. It's a very exciting project, I think, in my view, and it will certainly mean that we can, at the end of the project, we can say a lot more about groundwaters and microplastics and what the potential impact of microplastics might be in these invisible but very important ecosystems. So please get in touch if you're interested and you think you can help. And thanks very much for listening. Thank you. Right, I finished. It's a bit difficult when there's no, hang on, wait a minute.
Thank you. Thank you very much. I think there's already a question or a hand up, but we're just going to hold all questions until we have the panel discussion, which will start at 11:25 for Nigeria, UK and Cameroon, and 10:25 for Ghana and Senegal. So we're going to move on to the next talk, and we're going to, please put your questions in the chat. Charlotte is going to be monitoring the chat, so all questions can go in there, or you can put your hand up during the Q&A session. We're just going to call on Benedicta now, who's going to now take us from groundwater and microplastics to pollution in Ghana. I don't know, Benedicta, if you are online. I did see you. Ah, there you are. Can you hear me?
Yes, I can hear you. Just
perfect.
Share my slide.
So just for a bit of context on how I have learned about the work that Benedicta does, is Elvis, who is, I think, your PhD student or a colleague of yours. You've published quite a few papers together. He is one of the PAN-microplastic co-founders, so with Athena, who is in Japan at the moment, and Fedara, who is in America at the moment, and Elvis, who's just finished his PhD from Australia. We joined together to start the PAN-microplastic network. So I think that is the connection. Hey, Benedicta.
Hi. Sure.
You're a bit soft. We can see your screen. I think you're just a bit soft with the sound. I don't know if anyone else is hearing.
Like, see this? I cannot see my screen, so.
No.
So can you see my screen from your end?
Yeah, so we can see your screen. It's not in slideshow mode. It's just, yes, there you go. It's in slideshow mode now. And Pete Collier has just said that it just sounds like we can hear you, but it's just a bit distant. So I don't know if it's
okay,
just.
Maybe I have to speak up a little more.
That's sounding better.
Yeah.
Awesome.
Good morning, everyone. I hope you can hear me a bit louder. So, yes, my name is Benedicta Fosu-Mensah. I'm with the Institute for Environment and Sanitation Studies, a senior research fellow there, University of Ghana. My background basically is natural science, and my research actually it's in environment and it's quite wide. So I do a couple of research in environmental quality analysis, so look at heavy metals, waste, pesticides, residues, also into climate change issue related, and then also into microplastics research. Also, for this presentation, I'll just give an overview of environmental pollution in Ghana. So just to give audience pollutions that Ghana is confronted with and what we are doing as researchers to really dive or research into some of these pollutions and its impact in the environment and also on human health. Yeah, pollution in general, or environmental pollution, is a problem in Ghana. And I will say that in many developing countries, pollution is becoming a very complex, fast-growing challenge in many developing countries. Environmental pollution is not just a health-related issue, but it's also a social issue, as pollution is seen to kind of cause people or destroy homes as well as communities or cities in general. So basically for this presentation, I'll look at three main environmental pollution that we are confronted with as a country. We have problem of e-waste. We have also pollution of water bodies as a result of illegal mining, and also microplastics. E-waste is fast-growing domestic problem, and it's a waste stream that is fueled as a result of high demand of electronic and electrical equipment. And this is also as a result of most often these equipments have very short lifespan. Currently, in West Africa, the informal recycling of e-waste is becoming an emerging problem, or is really becoming a problem. And in West Africa, Nigeria and Ghana are known as the main hub or main source of imports to the West African region. Most often in e-waste, the recycling involves melting of the electronic boards, and this is most often done in open fire. And this is done to extract valuable metals and also cables that are in these gadgets. And the idea is to recover the copper from these waste. This method of recycling e-waste have resulted in emission of high concentration of toxins to the atmosphere. And also these toxins find their ways into the environment. This pollution or emission of these toxins affects the workers at the site as well as residents that lives around the area that this activity takes place, as well as the communities in general. So in Ghana, this is a common site that you find in e-waste areas. So you have young guys having wheelbarrows, collecting this e-waste from the length and breadth of the country into a particular area that we refer to as Aboboshi. That is the hub of the e-waste recycling. And so you see lots of these young guys going around from either homes and collecting these gadgets in order to retrieve the valuable components. When these gadgets are brought to the site, various tools, as we can see, are used to dismantle those gadgets into the different parts. And then the various valuable parts are then retrieved. And most often you will see these young men burning or using open burning to retrieve the valuable metals or components of these electronic waste. So we can see from the picture a young man trying to burn cables and then retrieve the copper that's in these cables. Most often they use car ties to carry out this. At the process of burning and retrieving these valuable elements or components, we have situations where certain pollutants or some pollutants are emitted into the environment. E-waste contain a number of metals, including gold, silver, platinum, mercury, lead, arsenic, and many other compounds that in many situations or many occasions are very toxic to organisms. During the process of dismantling, burning, and refurbishment, these toxins goes into the environment and thus pollutes water and the air. The negative effect of heavy metals as we are exposed to has health implications, including liver and kidney dysfunctions. I have carried out a number of research in that area, looking at the impact of e-waste on the environment and also on human health. These are just a few articles that I've published in relation to this area. Another challenge that Ghana is faced with has to do with illegal mining, what we popularly refer to as galamsey. And this illegal mining basically means that mining without license or permit. Though it is illegal, but it's estimated to employ about 1 million people in Ghana and accounts for about 35% of total gold production in Ghana. The activity of these illegal miners are mostly done along rivers and terraces, as well as along streams and also in low-lying areas. There's been a lot of legislature and a lot of laws put in place to regulate or to stop people from engaging in these illegal activity. But these practices still persist, and this is quite a serious issue in Ghana currently because of the level of environmental degradation as a result of the activities that is being done by those people. There are a lot of waste that is generated as a result of mining, and most often this waste is discharged into the surrounding environment, especially water bodies, without the recourse to protecting the environment. When you visit most illegal mining sites, this is what you see. A lot of young men in groups with champagne and smaller tools digging the ground without any procedure in search for gold and other minerals. Those have caused a lot of environmental degradation, a lot of biodiversity loss, and a lot of pollution to our water bodies. I would say that currently there's really been a lot of effort, including even organizing task force with military and police going around trying to stop these activities. But nevertheless, the practice is still ongoing. This is a common sight, or this is what you see of one's very beautiful river known as River Pearl. Before the activity, I would say in 2014 thereabout, this is how the river looks like, and currently this is how it looks like. The picture to my left, this is the current state of once beautiful river that we have in the country as a result of illegal. Again, we've seen a lot of destruction of arable lands and also the environment as a result of illegal mining. Sadly enough, we've had situations where even cocoa plantations are totally destroyed as a result of illegal mining. In most cases, the farmers that go into plantation or cocoa farming are not necessarily the owners of the land. The lands are sometimes leased to these farmers. And when miners come around, there are some chiefs and some opinion leaders that release this land to these illegal miners. And they go carrying out these activities without even informing farmers. So we've had farmers losing their plantation as a result of illegal mining. This activity over the years has caused a lot of destruction to our biodiversity, arable lands, forests, and also the resources in the forest. The activities have also resulted in pollution of soil and also groundwater as well as surface water. There's been a lot of research into impact of mining in our environment, including trying to find the various concentration levels of some heavy metals and also the health risk associated with illegal mining. And results shows that we are seeing an increase in the levels of these heavy metals. And even with the health risk analysis that we've done, shows that people involved in illegal mining are exposed to some hazardous chemicals that is resulting in an increase in the cases of liver, kidney, and also some other skin leeches, even in our hospitals within the communities that these activities are taking place. Another emerging issue that Ghana is faced with has to do with plastics. I would say that over the years, there's been a number of debates about banning of plastics because of the level of devastation, the level of pollution of this material in our environment. Currently, I would say that this discussion is more at a political level because various political parties have some kind of fear of the implication of banning plastics or some plastics, especially those that do not easily degrade, banning them from being imported into the country because of the implication of that on employment, and for that matter, votes that political party may get as a result of that policy. Globally, I would say that plastics is becoming an issue and becoming an environmental challenge. In developing countries, it's even becoming more serious because of the volumes that we get each year and the volumes that are released into the environment. Globally, we have about 2 billion tons of municipal waste, out of which 33% of this waste is not properly managed. These waste that are not managed most often are left or disposed of in the open dump site. And the situation is even worse when it comes to developing countries, with Ghana inclusive. In most developing countries, and Ghana inclusive, 93% of waste is normally disposed of in the open dump site. And out of these, 12% form part of the municipal waste that is. These wastes that are disposed of with plastics inclusive makes this plastics to litter the environment. And finally, these plastics find their ways into aquatic environment. Solid management in general is a major challenge in Ghana. And most often, just about half of the waste that is generated is collected and often dumped at open dump sites and at landfills. Little is done in Ghana when it comes to plastic recycling, though we import a lot of plastics into the country, but recycling is very. In Ghana, annually it's estimated that 2.5 metric tons of plastic is imported, and 73% end up as waste material. Only 19% of this plastic is reused, and 0.1% of it is recycled. So this shows that we have a lot of plastics that are disposed of most often in open dump sites and landfills. The types of plastics that most often are imported includes the PVC, the PCs, the PP, and then the PEs. Again, the sources from which plastics are found in the environment include the municipal, the industrial, and then the commercial sources. Whilst municipal plastic waste is seen as the most common source of plastic waste. This is because almost everything that you buy in Ghana, no matter how little it is put in plastic for the customer. So these leave huge volumes of plastics disposed of on daily basis. And most often, because of the improper management or collection of waste, a lot of this plastics find its ways into the open environment and also into gutters as well as the streets. Sadly enough, this is the site that, or this is what you see in some beaches or in some areas within Ghana because of the volumes of plastics that are not properly managed or collected. And so once it litters and it rains, the runoff carry these plastics into the environment. And some are washed to our seas, and sometimes the waves carry them back to the shore. The situation is quite worse. It's worse during the rainy season, also the runoff. Now, looking at Ghana, we are located around the Gulf of Guinea, and we have a lot of lagoons that are open and also closed. So there are times around all the lagoons that are located in areas that we have communities around. sadly enough, this is the site, or this is what you see within these various lagoons that are located in different parts of the country. For plastics, there are different types of plastics. We have the large, we have the small, and once these plastics are washed into the environment and find their ways into the lagoons, the sea, and rivers, we have situation where these plastics, that's the large ones, get the aquatic organisms are entangled, and these have implication on the survival of these aquatic organisms. We also have situation where organisms or aquatic organisms are not able to differentiate between food and microplastics or plastics in general, and so we sometimes mistaken this plastic as food and inject them. There are also some effects of these plastics as we get into the environment, including toxicity of the liver, that's aquatic organisms, and then also causing endocrine disruption. Currently, I'm involved in a project that looks at microplastics and plastics derivative chemical contaminants in Africa, implication on human health, and the loss of aquatic biodiversity. This project is funded by Jennifer Waltz, and our PI, who is currently with us, is Dr. Gideon. I would say that this project is a collaborative project involving a number of countries that I'm the lead for Ghana. There are a number of objectives for this project. I know Dr. Gideon will give a more detail about the project, but for Ghana, what I'm looking at is to investigate the presence of microplastic pollutants in freshwater and marine systems. And then to also look at the presence of various plastics derivative endocrine disrupting compounds, and to determine factors responsible for retention of the different microplastics and the EDC. These are some references that I looked at. Thank you very much for attention. Thank you.
Thank you so much, Benedicta. That was really, really a great overview of the pollution issues in Ghana, and nice to see Gideon's project is starting and things like that. And hi, Gideon, I did see you log on, so it's nice to see you. But yeah, let's get some questions in the chat and write some questions down for the panel discussion that will happen after Andrew Joseph's talk. So now we're going to move on to Andrew's talk, which he's going to be talking about antimicrobial resistance, but I won't speak too much about it. I'll let him introduce himself and the topic before I say something that is probably incorrect. Thanks for joining us, Andrew. That looked good. I don't know if we can't hear you, Andrew. I don't know if you've started speaking or not. Just wanted to make sure.
Okay, all right, thank you. Okay.
No worries.
So greetings to you all. Karibu to anyone from East Africa, and Akwaba to those of you present from Ghana in the West. I haven't forgotten about my friends in Nigeria. I'm yet to visit and do some amazing science and also learn the culture. In this presentation, I will be discussing AMR, providing you with an overview of the FAO Reference Centre and our engagement with a specific focus on West Africa. First and foremost, I'd like to give a shout out to the Ref Centre team for some of the work I'm going to present. So AMR, antimicrobial resistance, which is often referred to as the silent pandemic, can be described as a microorganism's ability to develop and maintain mechanisms to disable the effectiveness of bioactive compounds that may otherwise have bactericidal or bacteriostatic effects. So these are compounds like antibiotics, antivirals, antifungals, and antiparasitics, to name a few. And if you haven't gathered by the title, AMR represents a major global threat to the One Health concept and the attainment of sustainable development goals. So currently, antibiotic-resistant bacteria are estimated to cause around 800,000 deaths globally per annum. And in the worst case scenario, by 2050, around 10 million people in the world will die every year from bacterial infections. This really poses a serious challenge to the functioning of healthcare systems and represents a high economic cost to society. You know, as we have witnessed with the COVID-19 pandemic. But anyways, at a global level, the environment is increasingly being recognised for the role it might play in the emergence and spread of AMR. And there are numerous calls for more action on many fronts. So the O'Neill review, which gave us those worrying estimates on death toll and economic cost to society, and in Africa, the African Union published a framework for the antimicrobial resistance control 2020 to 2025. And also on on higher levels, like within the OIE and other and also UN entities such as the FAO, WHO, and the UN Environmental Assembly, that they've all reached a consensus to advocate strategic action for environmental surveillance to identify knowledge gaps and further understanding on antimicrobial contamination in order to develop robust action plans to mitigate the spread of AMR. So the determination of environmental contamination levels by the resistance driving pollutants is fundamental to improve the current state of knowledge on their source pathways, transport, fate, and resulting ecological effects. So as you may know, one of the key contributing factors to the emergence and spread of antimicrobial resistance is the inappropriate use of antibiotics in human health, food and animal production systems, and also crop farming. This has a direct impact on the aquatic environment through the poor management of waste coming from households, farms, factories, and human and veterinary health settings. So although widespread AMR has been mostly attributed to the selective pressures generated by inappropriate use of these antimicrobials, there has been concerns raised based on recent growing evidence regarding co-selection for AMR among bacteria exposed to non-antibiotic compounds. Therefore, in the context of marine pollution, understanding the dynamics and behaviors of these resistant driving pollutants in rivers, estuaries, and coastal waters, and how they contribute to the emergence and spread of resistant bacteria is of real importance, not only to human and animal health, but also the ecological health of the aquatic environment. And I believe more research using a synergized approach is required, as there are other chemicals such as metals and biocides that select for resistance genes. And also little is known about the prevalence and implication of these resistant genes in the food web of fish-eating organisms. As I just mentioned in the previous slide, co-selection of genes that are conferred for resistance to chemical hazards is of potential ecological and clinical importance. Co-selection can be broken down into three mechanisms. So you have co-resistance, which occurs when resistance genes are located on the same mobile elements. So, for example, your plasmids and integrons. And how this works is that bacteria in the presence of minimal selective concentrations of metals, for example, may select for antimicrobial resistant genes, and vice versa. You also have a cross resistance, whereby one resistance gene can offer protection to multiple hazardous chemicals, and through selecting for genes that confer the use of for efflux pumps. So these are seen in some of these examples here, where some of these compounds have demonstrated the ability to share structural and functional mechanisms of resistance, such as reduced membrane permeability, compound alteration, and the downregulation of porins, or as I just mentioned, the use of efflux pumps. The main point here is that this issue of resistant driving pollutants, coupled with the bacteria's mechanism of horizontal gene transfer and acquisition of mobile elements, is a major concern for the global effort to reduce the spread of AMR. This concern needs to be really addressed using a One Health and interdisciplinary approach to study the interrelatedness of such pollutants, the emergence of AMR genes, and the overall contribution to AMR. In this slide, I'd like to briefly highlight some of the work we have done within the marine environment space as a case study. So this is a baseline AMR monitoring study that was initiated in a number of Gulf Cooperation Council countries. The objective of this study was to develop and implement a regional baseline monitoring survey of the presence of AMR in coastal marine environments. This was done through the analysis of seawater collected from high-risk areas across the 4 countries. E. coli strains were used in this study as an indicator organism, and 560 isolates were analyzed as part of this monitoring program. Multi-drug resistance and reduced susceptibility to critically important antibiotics were observed, and a subset of isolates were also genome sequenced, and a high carriage rate of quinolone resistance and extended beta-lactamase resistance genes were observed, which correlated with the reduced susceptibility to fluoroquinolones and third and fourth generation cephalosporins. This is also interesting, as several other surveys have shown a strong association between quinolone resistant genes and ESBL producing Enterobacteriaceae, and these genes are thought to be co-located on the same plasmid. So this is actually an example of co-selection. Anyways, this study is important because of the resistance patterns that were observed. There's demonstrated applications of both phenotypic tests and genotypic-based approaches, and the overall establishment of a transnational AMR surveillance framework focused on coastal and marine environments. All this is useful data for generating evidence-based policy for development of robust environmental management interventions. Now just to give you a bit on on who we are and what we do. So we're the UK FAO Reference Centre for Antimicrobial Resistance, and it is formed by basically 3 executive agencies of DEFRA. So you have APHA, which focuses on terrestrial animals and the environment and food. You have CFAS, which focuses on aquatic animals and the environment. And then you have a VMD, which focuses more on the sort of antimicrobial usage and formulation of policy. We were designated a reference center by the Food and Agriculture Organization of the UN in April of 2019. As a reference center, we employ a One Health approach to our work, recognizing that the health of humans, animals, and the environment are all interlinked, and we try to ensure that that in our work that each dimension is considered when developing these programs of work in order to achieve impactful outcomes. This One Health approach is also useful in various areas such as zoonosis, food safety, and environmental and climate change, just to name a few. So the purpose of the reference center is to gather together the shared expertise of DEFRA agencies to fulfill our mandate to provide high quality science and policy expertise within the global community to specifically tackle AMR in terrestrial and aquatic animals and the environment. And we have various mechanisms of doing this, but just to name a few, we do this by supporting the implementation of national action plans on AMR, by also supporting the development of scientific capabilities and quality management for research and surveillance of AMR and antimicrobial usage. And through our colleagues, VMD colleagues, by providing advice and support for formulation of policy regarding AMR and antimicrobial usage. So the Ref Centre was formally announced in November of 2019 in Accra, in Ghana, and we immediately began conversations with competent authorities to try and establish the best way to really try and implement, help them with the implementation of the veterinary and aquatic health aspects of their AMR national action plans. We have also, in 2019, we also went out there to visit more stakeholders and get a better understanding of the lay of the land. And we also conducted a workshop and a training course out there. So speaking to people from within the different people within the sector, the terrestrial and aquatic animal farming sectors, and basically just a little bit of antimicrobial stewardship work, and also with scientists in the lab, teaching them some of the antimicrobial susceptibility testing techniques. We also hosted 2 Fleming Fund Fellows at the Weymouth Lab, and I believe they may have also gone to APHA to look at the facilities and also just to get an understanding of our labs and how we function, and what they could take back for that from us to their labs to ensure that they can implement good quality management procedures within their own lab spaces. Currently, we're also supporting a newly appointed Fleming Fund Fellow from the Fisheries Commission, and we're actually going out there next month to assist him in conducting a survey regarding antimicrobial resistance and usage in tilapia and catfish production systems. Again, with Nigeria, just before the pandemic happened, we welcomed delegates from the Federal Department of Veterinary and Pest Control Services at the Weymouth Lab, and also at APHA Weybridge. And we're currently mentoring an aquaculture Fleming Fellow. And we recently conducted helped them with the planning of a AMR surveillance project focused on pig farms and the impact on the environment around the farm. We try to do most of our work in close collaboration with the UK Health Security Agency to ensure a One Health approach, and this helps in our aspirations of becoming a UK One Health reference centre. And on top of all of that, we have trained 132 scientists from government, academic, and also private labs within Nigeria. So we're very much active within the West Africa scene, and we're happy and open to collaborate and assist where we can in regards to AMR. So I hope this presentation has touched on the importance of addressing AMR from a marine pollution perspective by highlighting the concerns of resistant driving pollutants and their overall contribution to the spread and emergence of AMR genes, and also demonstrated the work we are doing as a reference centre to assist competent authorities within these African countries with the implementation of their AMR national action plans through undertaking capacity building activities. Thank you for listening.
Thank you so much, Andrew. I'm gonna now pass over to the Q&A session, which will be led by Charlotte. I was just wondering if the three speakers could put their cameras on and see if we can put you in a panel view. And then please, audience, bring your questions in. You can type them in the chat, or you can put your hand up, and we've got about 20 minutes for questions. You are able to put your hand up, and then we'll be able to unmute you, or you can unmute yourself and you can ask the question. And if you can, just introduce yourself and who you want to ask the question to.
Holly, can I just say, it's Anne here. Can I just say I can't put my camera on? Actually, my computer is denying the fact that I have a camera, which is not true, but there we are.
No worries, no worries. But yeah, I don't know, Benedicta and Andrew, you're welcome to as well. But if it's going to cause everything to crash, then maybe we should keep things off. We'll just look at you, Andrew. I don't know, Benedicta, if you could put your camera on at all. We do have, I don't know, Charlotte, if you want to, I don't want to take over, if you want to take over from here.
Yeah, I'm going to leave my camera off because it's not working at the moment. I also have bad internet connection. In the chat, we have got a question from Pete Randall. Pete, do you want to ask your question to Benedicta?
Yeah, sure, can do. Yeah, it's just with your presentation, you obviously showed some images there of turtle entanglement. So I just wondered if there's any issue within Ghana with sea-based sources of marine litter, such as abandoned, lost and discarded fishing gear, and whether you have any feeling as to whether that is a within country problem or also an issue with other countries' vessels as well, transboundary.
Yeah, well, I will say that with the issue of entanglement, we have a similar situation in Ghana. There are other publications or some publications that have shown that there is some level of microplastic pollution, and some of these pollutants have been found in the muzzles of some fish. So of course, what I show is not, it's, yes, we have a cross-boundary country challenge, but it's also something that is to some level found here in Ghana. But I'm sure that I would have a more definite finding with this current project that we are running. I've just gone to the field, we've taken the samples, and we are doing some extraction. So very soon we would analyze the samples to get more detailed results from the samples that is being collected. Thank you.
Thanks very much.
So yeah, next in the chat we have a question again for Benedicta from Modu. Would you like to ask your question, or would you like to read it out? Okay, so I'll read it out. I'm not sure if it's a question or more of a statement. It says, as plastics are used by almost all Africans, regardless of socioeconomic status, plastic pollution and its widespread effects are relatively increasing day in, day out. Research is the only way to understand this problem and prefer solutions. Are these funds specifically targeted at supporting such activities on the African continent, especially to support young researchers with relevant ideas? Benedicta, did you have a response to that? Sorry, the question was aimed at you.
Yes, well, the grant is not targeting PhD students. I would say it's quite a small grant, but it's first to establish the levels of these pollutants in aquatic environment or aquatic ecosystem. And based on that, we could make some recommendation and also some advocacy as well as education. Currently in Ghana, there's a lot of education and advocacy ongoing about microplastics and its implications or its impacts on our health as well as the aquatic ecosystem. So the outcome of this research would kind of serve as a further emphasis on the levels of pollutants as far as microplastics is concerned in our environment, especially the aquatic environment, and the implication of this for aquatic organisms as well as human health, because once it gets into the muscles of these organisms or gets into these organisms that human consume, what happens is that we are, by accumulating these pollutants in our system, and it has some health implication. So yes, the outcome would serve as a more emphasis or a more evidence of our activities, the impact of our activity in the environment, and also suggest some measures to stop these menaces in our environment. Thank you.
Yeah. Next question is, I'm sorry if I pronounced your name wrong, Kwaku. Would you like to ask Benedicta?
Okay, thank you for the opportunity. I'm Kwaku Diodu. I'm a veterinarian working with the Fisheries Commission. I want to ask Benedicta whether the research also involves the Volta Lake or it's just the marine environment.
Okay, thank you. It also involved the Volta Lake. So for the sampling, we took samples stretching from Sogakope all the way towards Mefe area. So it covers voltaic as well, because we know there's a lot of economic activities going on along the lake, and also you can visibly see plastics along the lake. So yes, the voltaic was also included in the sampling.
Okay, that's great. I would like to suggest there is this area called Gemini Market around the Volta Lake. There's a lot of marketing activities that go on there with a lot of plastic pollution. I'm just suggesting if you can consider that area, because they're going to see a lot of plastic pollution along that stretch. So it's just a suggestion for consideration. Yeah. Thank you.
Okay. Thank you. Well noted.
Right.
Thank you.
Yeah, next person with a hand up is Holly.
Hey, I just said I wanted to also respond to Modu's comment about funding, and it's something that I think we should all be discussing potentially in tomorrow's more discussion-focused workshop. Is really about, we understand what the data gaps are, but sustainable funding is really important, not just in Africa, but across the globe, especially if we want to see if interventions are actually going to be successful. We really do need to take the burden off researchers and parties that are gathering this data, so that is not an issue every year trying to find that source of funding, but data is gathered and implemented and fed back to decision makers and stored in a repository. It was something that we discussed quite a bit at the marine waste management the conference in South Africa a month ago. So yeah, something to think about.
Yeah, and there is just, because no more hands, in the chat, Holly did ask a question to Anne about the geographical spread of samples and partners, and do you already have them? Anne responded in the chat saying that there's none in Africa, lots in Europe, a sprinkling through Asia, and that Australia and New Zealand are also covered. Andrew, your hand's up.
Hi, I just want to ask about the microplastic sampling, and whether an integrated sampling approach has been used within this survey. Maybe looking at some of the microbial communities present within these plastic samples, as I'm aware that they harbour biofilms and could be vectors for AMR, spread of AMR resistance genes. So I just wanted to know if there's any sort of microbial work going on with these samples.
Hi, it's Anne here. Yes, there is microbial work going on in the sense that in the other objectives, in the other work packages, we will be, let's say, enumerating, counting the microbes as part of the food web. We don't have planned any work looking at the possibility of the microbes acting as sort of like vectors, really. And the whole AMR thing is not something that we're covering, although I have to say that, I completely understand your interest in this. I think it's a really, really, really important topic, and it would be great to be able to piggyback something on, but it's not something that we're doing at the moment. Does that make sense? Yeah.
Yeah, no, that does make sense. Thank you.
Yeah. Actually, for the global sampling, we're not. We decided, well, originally we did plan to do the whole food web thing globally, but it was just so complex in terms of getting permissions for sending materials, biological materials, if you like, that we gave up on that. Although we do plan to do a subset of food web, and that includes the microbes, of course, with selected partners who. And the plan would be that we did it at their institutions rather than bringing material out and back to the UK, simply because of the issues involved with sending stuff with couriers and things like that with biological materials.
Right, thank you.
Okay. I also meant to say I was just rereading my reply to Holly, and we have a lot of people in the States as well. I forgot the rather large United States and Canada contingent there, but there we are.
Yeah, Holly, off you go.
Sorry, I actually had a question, and excuse my ignorance here, Andrew, but with this antimicrobial resistance, the sources of these antimicrobes and antibiotics obviously vary with different countries and across the globe. What are these sources and how do they vary? Because I guess something that we often trying to think about is, and why we wanted this workshop to be about pollution, is really to think about the fact that we have limited resources across the globe, and are these sources similar? Are we actually finding spikes and indicators that could be coming from similar industries, sectors, things like that? And are there ways, like you said in your talk, a lot of things we need to actually look at these kind of the synergy between various pollutants and the synergy between, and I think Benedicta was also talking about, you know, the effect of these as a combined contaminant.
Yeah, so you know, in terms of you know, looking at the sources, I would say it's mostly attributed to the technologies that current wastewater treatment plants have. So a lot of them do not have technologies to remove antibiotics from from incoming wastewater, and to even go back further, these antimicrobial compounds get into the wastewater because basically, you know, around 60 to 80% of antimicrobials are metabolized. So a lot of these compounds end up in wastewater as actual active antibiotics or byproducts of the antibiotics, so they're metabolites. So in terms of you know, the concentrations varying in different environments, I think that could be attributed to a lot of different things in regards to the practices that are occurring within that space. So, for example, from you know, coastlines where there may be or riverbanks, just off of riverbanks where there may be farming ongoing. Some of the land could be layered with manure or sludge, and these can end up into the environment through runoff and things like this. So there's, you know, I would say the concentration and sources of these antibiotics are attributed to different practices that may be ongoing within particular environments, if that makes sense. David.
Yeah. Hi everybody. I work with Andy on the reference centre, and actually that's a really good answer, Andy. Thank you very much. I think it's just also worth everyone to bear in mind that when we talk about AMR pollution, you can probably divide. It's actually probably both the selective agents, the antimicrobials and biocides and other things. We talked about co-selection, potentially important, but also the antimicrobial resistant organisms and the genes they carry can also be looked upon as kind of pollutants, I suppose. It's kind of complex because most AMR resistance genes evolve initially as part of the normal. That's how bugs get about and how they survive, is by producing these things anyway, from fungi and whatever. So they're natural parts of microbiota that often they get mobilized into pathogens and then can be transferred. So you can look upon as a kind of pollutants. You can look upon the resistance genes themselves can be pollutants. You know, the plasmids that are held on and that thing. So yeah, it's looking at it in this sort of kind of holistic way. And related to the first part is often where you have big concentrations or big impacts, where there's you know, either treated or untreated fecal waste. There's a key source of both those chemicals and also potentially resistant organisms and genes as well. So yeah,
yeah,
plenty
and
to be done for sure. Thank you.
Yeah. And it's just also worth noting that you know, some of these concentrations of these resistant driving pollutants are at levels that potentially have no effects, but you know, combined together, the synergistic effects can select for resistance genes, you know, whether that's metal resistance genes or antibiotic resistance genes, or you know, resistance genes for biocides. So I think a little bit more work is needed in regards to understanding the synergistic effects of these resistant driving pollutants on the emergence of AMR genes, and then also looking at the prevalence of these different genes within the environment and also the food web.
Yeah, thanks very much. Does anyone have any further questions at this time? I can't see any hands, but if anything does spring to mind, we can perhaps address it later. So for now, I'll just pass back to Holly.
Thank you very much. And thank you for the three speakers. I did actually have one last question, seeing we have a bit of time, for Anne. Because my background is microplastics, and I know a lot of the people on the call have a background in microplastics. A lot of the time we think about they're there, you know, are they an issue? Are they toxic? You know, the stuff that we're doing in the lab, we use very high concentrations to see if they have effect on organisms, and you mentioned this legacy effect of groundwaters. Where really, you know, we could stop using plastics, and all the plastics there could break down and eventually seep into these groundwaters where we can't actually remove them. Is that a major concern? Because I guess at this point in time, they're really not at concentrations we should be worried about.
Mm-hmm. Well, yeah, that's a really interesting point, as a matter of fact. And the answer is we simply don't know. I mean, I think the feeling, and it can't be more than a feeling at this stage because we don't really have data, is that given the quantities that there are mismanaged waste there is on the surface, you know, a proportion of that will undoubtedly make its way down into groundwater. But what proportion, and you know, with what rapidity, you know, how quickly, I just don't know. I mean, you know, I think it's a bit, as I said very briefly, it's a bit like the nitrates, really. You know, that is a very slow burn, a very slow build up, if you like, of concentrations in groundwater, but nevertheless potentially quite serious. And I guess that microplastics, and perhaps particularly nanoplastics, you know, there could well be a problem in the future in groundwaters, but we just don't know, to be honest. And that, in a way, that's what this project that I'm leading is about, is trying to get some sort of baseline information as to what might be happening now, and perhaps model potentially what could happen in the future. Yeah, I mean, given the importance of groundwater to you know, a lot of people seem to be talking about groundwater as a mechanism to develop resilience, if you like, to climate change. And you know, that to me makes it even more important that we find out, you know, what potentially the impacts of microplastics might be. Yeah, but this is very much I wouldn't say it was a fishing exercise exactly, but it's very much a start to the process.
Great, thank you. Any last, we've got a few minutes for any questions. Oh, Eric, can I unmute you? And hi, Benedicta, we can see you now. Eric, I don't know if you want to unmute yourself. Doesn't seem to be. If you can't unmute yourself, you might want to just put your question in the chat. I can't even unmute you, sorry. So, well, why don't we take our tea break now? If you have any questions, we can always put them in the chat and send them through to the speakers if they're not here at the end of the day. But we will have another panel discussion at the end of the day, and maybe a little bit of time left over, because I've left a little bit of time for any of that. Oh, we have another hand. Oh, we've got 1 minute now. If you can unmute yourself, you can ask a question. There you go. Hello.
Hello, good day. How are you doing?
Great.
Hello.
Hello, we can hear you. Do you want to introduce yourself and then ask your
Okay.
question?
Okay. My question is this. We've not started antimicrobial resistance surveillance in Nigeria. So we're expecting Dr. David Vanner and his team, so that we would like to know when they actually want to come in. Because since some of the letters have not brought the results that we expected, and we are delighted to have him. Anytime he will come and start working with us.
Thank you very much, Dr. Oganoiki. Just for everyone's benefit, she sort of leads on aquaculture health policy in Nigeria, and it's a privilege to have her on the call. So thank you very much, Dr. Oganoiki. And yes, we do need to sort out the paperwork so we can work properly with you guys. But yes, it's very much the case. And I think Suliat's on the call too. Our Fleming Fellow we're mentoring from Dr. Oganoiki's team too. So it's really good to have you guys on the call here today. Thank you.
Thank you very much. And yeah, paperwork is always a nightmare.
Thank you. Thank you.
So we're going to, if you have any other questions, put them in the chat. We're going to take a 15-minute break. I would suggest just muting yourself and your cameras and going off instead of exiting and re-entering, because I still haven't figured out how to not have the names come up, but hopefully in the tea break I'll figure that out. And yeah, we will reconvene after the tea break, which will be at 9. Yes, it will be at 12 o'clock. It's a quick tea break. So it will just be about 12 o'clock. And then, so that's 12 o'clock UK, Cameroon, Nigeria time, and 11 o'clock Ghana, Senegal time. So quickly grab some water, and then we'll have the next 3 talks, and then you'll be able to head off and do the rest of the work you need to do. See you in a bit.
Yeah, see you.
Bye.
Hopefully everyone is back or starting to make their way back. Had a stretch, had some water, has got a cup of tea. We have got 3 more talks. One is recorded. I cannot see, so it looks like the third speaker is not online yet, so I have sent him a message and hopefully he will. If not, that's more time for questions, but it would be nice to hear more about pollution and plastic waste in Senegal. But we do have a talk coming up from Patricia, who's going to talk about pollution in Cameroon. So I don't know if everyone is back, if anyone could maybe raise their hands as a reaction so I can see that I am not talking to an empty room and you're all still making your coffee. Okay, thanks, Patricia. Awesome, I see a couple, So I don't know, Andy, I don't know if you would like to put your camera on. Yes. Hello. Sorry, I put you on the spot. Seeing you know Patricia very well, I thought maybe you could introduce Patricia. We do have a bit of time, so we can, we've got a bit of wiggle room, so we can wait a few minutes, or you can do an introduction now, and then we'll see how everyone's joining. Up to you.
I'm happy to do either. I would say knowing Patricia very well is a stretch. Knowing her for a very long time is probably more accurate. But yeah, I'll do a brief introduction, and I hope I do you justice, Pat. So Patricia has joined us at quite late notice to step in for somebody else who couldn't do their talk. So we're very, very grateful for her for making herself available to do a talk that fits in with the theme of what we're discussing today. So I met Patricia a long time ago when she came over to the UK to work with University of East Anglia and came and did some work at CFAS. And I'm an ecotoxicologist and she works in ecotoxicology. So that's how our paths crossed. She came and did a bit of work in our lab and produced quite a few papers during her time in the UK. One of those was working on a multibacterial test system for looking at toxic impacts of herbicides and potentially looking at mode of action by looking at which bacteria were impacted and which ones weren't. It's a fascinating little test, and it was a test that was later picked up by part of the UK government to use for testing effluents from the oil and gas industry offshore, and was used as part of the assessment process to see whether or not these were going to have restrictions placed on them or not. So it was an important piece of work. And then later on, we were both active members of CTAC, which is the sort of professional body for environmental toxicologists and chemists. And I know that Pat rose to become the president of CTAC Africa and led CTAC Africa out to become a separate geographical unit during her presidency. So that's quite an achievement, and recognizing the size and depth of work that was going on and has been going on in Africa for a long time. And currently she's working as an associate professor at, and I'm going to really mangle the pronunciation here, so I apologize, but the Université de Chang in Cameroon. And I think that's the end of my introduction, and hopefully Pat is ready to do her talk.
I don't know, Pat, just let me know if you can't share your screen.
Thank you very much, Andy. I'm trying to find my presentation, which I think
we can see that.
Thank you very much, Andy. I hope you can see my screen now.
Yes, we can.
Just trying to get into slide mode. Yeah. Okay. Once again, thank you very much, Andy, and thank you for inviting me to this very important workshop. I'm really glad to be part of it. It was short notice, but I really appreciate the invitation, and I'm happy I'm participating. I found the 3 previous speakers' presentation very interesting. Andy has already introduced me. I would just add that I'm a founding member of the International Board for Environmental Risk Assessors, which was actually established by CITAC, and I'm bringing this up because we are actually organizing our first certification exam. Unfortunately, the applications just closed, but for those who might be interested in getting certification as risk assessors, we could talk about this later. Right. I am happy that Holly suggested that I should focus on some ecotox. Otherwise, almost everything I should have said on the overview of pollution in Cameroon would have been like repeating Benedicta's presentation. A lot of what I'm going to say on the overview would be very similar to what Benedicta presented already. So I will say something on the overview of the pollution in Cameroon, then get into the ecotox and risk assessment, and then some thoughts at the end. Well, as an introduction, we all know that pollution is a global problem, and it's a silent killer of millions in poor countries, because there is hardly any data on most of what is going on, and most of the known causes of pollution globally are presented here, which I don't need to repeat. And Cameroon, like most other African countries, have increasing environmental pollution problems. And so these are some of the important types or sources of pollution in Cameroon that I'll just give an overview about. That is agricultural.
Sorry to interrupt, Patricia. I don't think it actually is on slides, because we're still on your first slide, or I don't know if that's just me, if anyone else is seeing all of the slides, just to make sure. Okay.
I'm just seeing the title slide as well at the moment.
Oh, all right. Probably my slides are on my screen is moving. I wonder why. Maybe Holly, can you, maybe I should stop sharing and then you share the slides?
Yeah, sure.
Because on my screen is moving.
No worries. Hopefully I will be able to do this.
Okay, I'm stopping.
Let's see. So that should be the presentation.
Yeah, we can see that.
Okay, let's go from beginning. Display.
Now we've got. Okay. We can see both screens. That's it. Yeah.
Right.
Good.
Yes. Okay. Well, sorry about that. I was actually moving normally on my screen. I wonder why it wasn't going through.
All right. Is that working now?
Yeah.
Right.
We're on slide 1.
Holly, does that mean that you would move through the slides as I talk?
Yes, I think that will have to.
Hello?
You'll have to let Holly know, and she'll click on to the next slide. Oh,
Right, okay. Please move on. I think we're done with the introduction. Right, so, okay. So what I was just saying is that I will just give an overview of the different pollution types and sources, and I wouldn't spend much time on this because a lot of what I'm going to say is very similar to what Benedicta presented. Please move on. Right, so that's too fast. Right, okay. This is just to give some statistics, just to indicate the seriousness of the pollution problem. And Douala happens to be our economic capital. That is where a lot of the economic activities take place, and is situated just next to the Atlantic coast, and that is where most of our industries are located. And one of the reasons for that is the apart from other economic reasons, is the ease of rejection of their wastes directly into the aquatic system, usually without treatment. So some numbers, like you can see, hundreds of thousands of tons of estimated for BOD and suspended matter in the marine environment in Cameroon. Okay, and this has been mostly attributed to the petroleum industries, as well as food processing and chemical industries. We also have liquid industrial waste, especially solvent waste, at about 84,000 metric tons per year. So we can see that many of the industries are not willing to treat their wastes before rejecting them, and there's poor enforcement of the regulation. So we find this waste in the environment. And to make things worse, the industrial zone is not distinguished from the residential zone. So where all these wastes are being ejected, that is where the residents are found. So a lot of the population is exposed to them. Move on, please. Right, so more data. We also have untreated ballast waters from ships, lots of it, getting into the marine environment. I've already mentioned the use of stream as waste by industries, but also by the residents, because untreated waste, emptying of the septic tanks, get into the streams which flow into the marine environment. And then the drainage system is generally poor. And in this particular town, Douala, which is a major economic city, as I said, it is very, the topography is really level. And so with poor drainage system, it leads to a lot of flooding. We also have significant pollution from oil drilling, refinery waste, oil spillages, gas and flaring. Not too far in the southwest region, that's in Limbe, where the oil refinery is, which is also located along the Atlantic coast. Move on, please. Now, I mentioned that agricultural pesticides is also a major problem, because we know that in our developed countries, agriculture is practiced by over 80% of the population. Although mostly is smallholder farms, but we also have plantations, and there's the dependence on pesticides for to reduce losses from pests and diseases. And we find, as you can see in these images, the farmers that use it are generally untrained. You see, they are not protected, and the pesticides are easily available to them. Okay, there's very little control on pesticides. So you find pesticide stores around the corner, and most often these farmers gain their advice on doses to use and which pesticides to use directly from the pesticide vendors. And after using, they just throw the containers, the empty containers, directly into the water bodies, which leads to potential risk of harmful effects occurring on the aquatic organisms and eventually to humans. Please move on. Another important source of pollution is e-waste. It's true that the situation in Cameroon is not as bad as in Ghana or in Nigeria, because Cameroon ranked 14, but it is still an important problem because we generate, for example, in 2019, we generated about 26 kilotons of e-waste. With about 1 kilogram per capita generation. E-waste is a global problem, so you have some statistics, some global statistics here, and most of these e-waste that are generated end up either in landfills or in poorly equipped recycling facilities, or mainly at informal recycling sites, as Benedicta also mentioned. Please move on. Mining is also a problem in Cameroon, just as in Ghana, with a lot of artisanal mining taking place. This image was taken in Cameroon, but it looks so similar to the one presented by Benedicta. So we see there are several environmental problems that are caused by this mining. Apart from the land and water pollution, we also have deforestation, land degradation, and all these open pits, as well as the health risks to those who are involved in the mining activities. Please move on. Plastics, which is kind of the main subject for this workshop. We can see it is also a problem in Cameroon. We can see actually like a sea of plastics here, with people going through, sometimes picking them up to put things and resell and expose others. But we can see how it causes blockages of the little drainage facilities that are available, and therefore where there is rainfall, it increases it makes the floods even deadlier. So we can see here that Cameroon produces the highest amount of plastic waste in Central Africa. And out of the about 600,000 tons of plastic waste that is generated, only 20% is recycled. So most of the rest ends up in the environment and is very poorly managed. Let's move on. Indoor and outdoor air pollution is also a big problem. And we see that the sources are from car exhaust, bike, in fact, automobile exhaust. We know that Cameroon, like in most other African countries, a lot of the energy that we rely on is on wood burning, but also burning of kerosene and other fossil fuels, which generate a lot of carbon dioxide. And then when it's enclosed environments like in the kitchen, that exposes, it's a risk factor for many diseases. The picture on the left is the recent accident that occurred at the oil refinery in Limbe, generating this thick amount of smoke into the atmosphere. And there's very little data on the levels of pollutants that are found in the atmosphere and that we are exposed to. Please move on. Open burning at the dump sites is also a common source of air pollutants. So with that general overview, I thought I should focus now on some cost-effective ecological ecological bioassays and some risk assessment approaches that we have used. Andy mentioned the microbial assay for risk assessment. I initially wanted to present to include that, but given the short time limit, I couldn't manage to. But that is one of the assays we have used. Recently, we have carried out earthworm bioaccumulation, growth, and reproduction bioassays to assess the effects of e-waste, which I'm going to present. I'll say do some presentation on mixture toxicity test. We know that in the environment, we are not just exposed to single toxic pollutants at a time. We are exposed to the mixture, and so assessing the effect of the mixture is becoming more and more important. And then we did some ecological risk assessment. So please move on. I'll talk on each of these one at a time. So I'll start with the earthworm bioassays. Please click again so that everything just shows up on the screen. Yeah, okay, thank you. Right, so I'll start with the earthworm bioassay. We decided to, we did a preliminary survey on the e-waste recycling sites, and we collected some soil samples from those sites just to find out how the toxicity using earthworms. And we used local earthworm, a local earthworm species, Alma nilotica, instead of the standard species that is normally found in the OECD tests. And so we developed the culturing method for these local earthworms in the laboratory and exposed them to a number of soil samples from various e-waste recycling sites, and we compared the effects. Well, we first of all wanted to see if the some metals in the earthworms in the soils, in the contaminated soils, were bioavailable that could be picked up by the earthworms. And so we exposed the earthworms in the laboratory and sampled the earthworms weekly, processed it for metal analysis, which was done by ICP-MS in a lab in Canada through our collaborators. Please move on. Right, and so I'll just give a highlight of some of the results we obtained. We found that, for example, during the exposure period, the earthworms bioaccumulated the 9 metals which were under study. Okay, these metals were bioaccumulated. We also gave a period of post-exposure period where these worms were taken out of the contaminated soil and put in clean soil to see if they're going to depurate, okay, eliminate the metals. And we found that they were eliminated to various extents. For mercury, very little. The rate of elimination was very slow. But for some others, like lead, there was fast elimination. Cobalt was eliminated also to a great extent. So we could see that the behavior of the different metals are different. But what we could say is that the metals were bioavailable for uptake by the earthworms, and the bioaccumulation factors were calculated, and we found that for mercury, even though the concentration of mercury in the soil was really small, but it had the highest bioaccumulation factor. This work is published, and the reference will be given at the end of the slide. So for anyone who wants to read more about it, please, next slide. Right, so we also assess, having seen that the earthworms bioaccumulated the metals, so we now try to see if this bioaccumulation was to toxic extent the metals were accumulated to an extent that could affect various life processes. So we did the growth and reproduction assays where the worms were exposed to the various contaminated soils, and they were sampled weekly for the growth test. Weekly, they were weighed weekly, and then for the reproduction test is at the end of the experimental period. The number of young were counted in each container. Please move on. Right, so these are the results from the growth test, and we can clearly see when we compare growth, the mean metal concentrations, first of all, in the soil, we could see that it was clearly higher in the contaminated soil than in the reference soil, which in black. Then when we look at the growth curve throughout the period, okay, this is throughout the exposure and post-exposure period. They were exposed for 21 days and later on for 14 days, they were taken out and kept in clean soils. So we could see that the first two curves are for reference soils. We could see that the growth of the earthworm was good, but in the contaminated soils, they were significantly lower, particularly during the exposure phase. But after the exposure phase, we started seeing some growth taking place, showing some kind of recovery, which varied at the different sites. And we found that it depended on the level of contamination of the site. Please move on. For the reproduction assays, remember I said we counted the number of youngs, and we can see that when we compare the number of youngs in the reference site, for example, 90.5 youngs per 10 worms in the reference site, as against just 8 youngs produced per 10 worm on average in the e-waste site. So we can see there was a clear effect on the reproduction of the earthworms exposed in the contaminated soil. Please move on. So I'm moving on to another bioassay, that's the mixture toxicity test. And these were carried out using local fish species, Oreochromis niloticus, fingerlings, which were exposed in a short-term, that is 96 hours, standard acute static aquatic tests in tanks as shown on the screen. And 3 pesticides were tested: dimethoate and organophosphate insecticide. We have cypermethrin and deltamethrin, both pyrethroid insecticides. They were exposed, first of all, as single pesticides to establish the dose response curves for each of these pesticides. Please, next slide. And then we went into binary mixtures of those 3 pesticides in two composite experimental design, okay, where in one of the design on the right, you have the NN design, where we had a fixed concentration of one pesticide and we varied the concentration of the second pesticide. And then in the ray design, we have a kind of equivalent concentrations of the 2 pesticides in the binary mixture. So as demonstrated here. Please move on. Now, the result, well, what we see here are the results for the single pesticide. So we see the dose response curves for the 3 different pesticides studied. So we could see that the toxicity of the different pesticides varied very widely. You can see for dimethoate, you needed a high concentration to cause harmful effect, while for permethrin and deltamethrin, we had to use a separate scale to plot on the same graph for comparison. But we can see here that the dose response curves were very similar, and therefore it was possible to merge these results to come out with a single dose response curve and to come up with an equation which describes this relationship. And this equation was then used to predict toxic effects of mixtures. This work is published and the reference is given for those who want to know more. Please move on to the next slide for us to see the mixture, the prediction. So just given here, we are seeing how the mixture effects were predicted. So using two models, the concentration, the independent action and the concentration addition models, which were used to predict the effects of the mixture compared to the observed effects. So what we can see here is that the predicted effect was much lower than what was observed, which means that the mixtures were not additive. The effect of the two pesticides in the mixture was not additive, but it was synergistic. That is, the effect was much higher than what could have been expected if it was based on concentration addition. Next slide, please. So looking at the ecological risk assessment, we were also able to carry out some risk assessment, pesticide risk assessment, using the primate model, which is a simple model. Primate means pesticide risk assessment to man, environment, and trade. The model, the website is given, was developed by Van den Brink et al. In the Netherlands, and it's freely available. It's developed with a focus to tropical areas, and the advantage of using this model is that it has very limited data requirements, because we know that we always have difficulties having data in the developing countries for risk assessment. And so it's important to be able to have a model that can at least screen the risk, the preliminary risk, to have an idea before moving on to higher tier risk assessment. So in this model, went to the field, we did some surveys, including questionnaires, to understand the pesticide application schemes used by the farmers. We did some measurements on nearby streams that were that were flowing by the farms, and some geometrical and physicochemical characteristics of the streams were measured. We also, through literature, collected the physicochemical and toxicity characteristics of the various pesticides that through our survey we could find that were being used. And all this information were introduced into the primate model, which is able to calculate the predicted effect concentration of the pesticides. Is also and is able to express able to also calculate the predicted no effect concentration, PNEC, and express the risk as exposure toxicity ratios. And there is a scale where if the exposure toxicity ratio is less than 1, the risk is acceptable. If it's between 1 and 100, there is probable risk. Remember, this is screening level. And then when it is higher than 100, the risk is definite. So please move on. Hello? Next slide, please. Thank you. So these are just highlights of the results from the primate risk assessment. So we found, we identified some insecticides and fungicides which pose definite risk. We can see here the associated, please move back. Well, we identified some pesticides that pose definite risk and some that pose possible risk. Some did not pose any risk. And what was interesting in this study was that most of these pesticides which posed a definite risk were actually applied at doses lower than the prescribed doses, and yet they still came out as posing a definite risk. So this work is also published and the reference given for those who want to know more. Next slide, please. Right. So, so those are the three tests I was able to put together within short notice, and I just thought I could conclude by giving some thoughts based on some suggestions by other authors also of why we are having this problem and what can be done about it, the pollution problems. And what we find is that in many cases, there are actually a lot of regulations and policies, laws already established, but they are not being properly implemented, if at all. And probably in most cases, there are financial difficulties. So possibly, the use of this low-cost toxicity test may encourage the introduction of monitoring programs. Also, another problem raised was the absence of target-oriented research, which I've highlighted here because I think that is what is most relevant to us. And we really need that communication between researchers and policymakers to be able to actually orientate our research to do things that are actually going to be implemented to improve on the pollution problem. Okay, there are also other problems where there are, in Cameroon in particular, there are some conflict of competence between various ministries. You find several ministries which are overseeing the same things, and sometimes there are conflicts on who has to do what. It's not clear. Right, so sometimes the existing legislation also may be inadequate or ill-adapted, and in some cases, the absence of national norms. So these are some of the things that I think we could discuss and see how we can contribute to improve the situation. Also, there's insufficient data. Next slide, please.
I don't think there is another slide. Oh, my bad. There is. Sorry. Can't find it. There you go.
Yeah, right. Yes, yes. Well, what I've said is covers this slide. So they need to educate the population, need to involve all the stakeholders concerned in any particular environment when planning or developing any activities. These are some of the solutions that have been proposed that I think maybe we can also incorporate. I think, yeah, so this is just acknowledging most of the students that have been involved in carrying out the research in the lab to give us this data. And then following this, you just have the references that have been used. Thank you very much.
Thank you so much. Oh, hopefully gosh, I've got so many screens and so many things. It's like it's the first time I've ever done a virtual workshop. Not that we've been doing this for the last 3 years. So thank you so much for that. And if you have any questions, please do put them in the chat. I think it's really given us a nice overview and a different viewpoint from what was discussed earlier today. And now we're looking more at these consequences and things like that. So please do put your questions in the chat, but we will have a Q&A session afterwards. We've got a recording now. So Sue Ware, who is part of CFAS, is part of the emergency response, does a lot of emergency response for various pollutants. I thought it would be a nice topic that we could listen to. Unfortunately, she was actually at a steering committee that she discusses in the talk, and then is on annual leave from tomorrow. So she kindly recorded the talk for us. So hopefully I'll be able to do this talk. In the meantime, let's see. If you could just bear with me, because now I've got a few of them open. Okay. Okay. Looks like I've managed it. Yes, you should be able to. You should be able to see the screen. And please do let me know if the sound is quite quiet. So if you could just raise it up on your side, because I think it is on max here. So yeah, I will let you.
Hello everyone, hope you're enjoying the conference. So I'm just going to spend a few moments
Holly, we might have lost the sound for everyone. We can see it, but we can't hear it.
I think if you click on the share button and you should see a little toggle that says include computer sound. If you press that, then it should play the sound of the presentation.
Okay, let me.
Yeah. Gotcha. Thanks for that.
So was it working for everyone else?
No, it wasn't working for me either.
Not for me, no.
No. All right. We tested this earlier, Charlotte.
It worked. It worked for the first few seconds, and then it went.
Exactly.
Andrew, what were you saying? We need to change you said the speaker view. So if I share.
Okay, so on your Teams platform, if you click the share button.
Yeah.
And then you should see at the top right, it says include computer sound.
Yeah, and then it says go to your device audio settings and select speaker. So
yeah,
I go that, and it's already on speaker.
You should just be able to click that, and once that's on, should be able to play the presentation with the sound.
Yeah, let's try one more time. Okay, hopefully this works. I've just unplugged my second screen, so I think maybe it was on different screens.
or sort of in the European context.
Yeah.
More recently,
working.
Our emergency response function, and that's in relation to pollution events that we respond to. So much of our work around emergency response originated from the development of the PREMIAM guidelines and the approach that we have developed under PREMIAM for post-spill monitoring and impact assessment. The guidelines were originally developed for response to UK incidents primarily, and they were really set up to cover the types of habitats and species that we may wish to monitor after a pollution event within the UK or sort of in the European context. But more recently, the guidelines have expanded to cover a sort of a wider geographical setting, and they've actually been really quite instrumental as part of our response to a number of international pollution incidents. PREMIAM really does just focus on the post-spill monitoring and assessment element of emergency response. It doesn't really go into some of the elements around immediate response. So, for example, best practice for cleanup or mitigating the impacts of a pollutant along the shoreline, for example. So this really does focus on the sort of subsequent stages that relate to really making a scientifically robust assessment of what the pollution impacts have been. So I'm just going to use a case study to illustrate some of the ways that the PREMIAM guidelines have been applied in a non-UK context. And this case study relates to the MV Wakashio. The Wakashio was is a bulk carrier that was grounded in 2020, sort of summertime, just off the coast of Mauritius. It was 1.5 kilometres off Point Desney, which was just to the southeast of the island. It was sailing empty. It wasn't an oil tanker. It was a bulk carrier, and it was sailing empty at the time. It was on its return voyage from Singapore to Brazil. It was carrying just under 4,000 tonnes of fuel oil and 200 tonnes of diesel. And when it ran aground on the 25th of July, it was during a pretty bad period of weather, and subsequently the vessel did break up and release a quantity of fuel oil into the environment. It was quite interesting in that we were dealing with a type of oil that was relatively unknown or less well understood than some of our more traditional fuel oils, but I'll come to that in a moment. But it's important to just understand how the knowledge that we've got of the particular substance in the environment, how it behaves, the properties, that's really fundamental to being able to track where it may have gone and also what impact it might have on the various receptors that it comes into contact with. So that's habitats or species or human health as well. So in the context of the Mauritius incident, the Wakashio grounding, we were able to contribute from a UK perspective to the emergency response that was offered by the international community. We were able to mobilise relatively quickly and put a team into the in-country to provide support to the Mauritius government and allow them to draw on our knowledge and experience, particularly around post-spill monitoring and assessment, to help mitigate the impacts of what had happened, but also understand what the impacts were where they had occurred. So we were able to put a number of experts into country. There was myself and two colleagues, Philippe Basuda, who was an analytical chemist, and also Michelle Devlin, who is an expert on all sort of elements of pollution, really, and also tropical ecology. So she has great experience from previous work on the Great Barrier Reef, for example, in relation to coral reef ecology and other tropical marine habitats. The support that we were able to offer was funded under the UK Aid programme, and our involvement was coordinated by DEFRA, the FCDO, so the Foreign Commonwealth Department, and also the British High Commission. So what did we find when we arrived in Mauritius? Well, we found that because of the time that had already elapsed since the vessel had broken apart and discharged the oil, there was a really effective cleanup operation underway, and it was being led by a number of professional cleanup companies. There were also a number of expert groups that had already started to think about the monitoring effort, and these were made up of in-country Mauritius experts and responsible authorities, but there was also quite a big contribution from other international teams that were there, sharing their knowledge across various elements of emergency response, again to help the Mauritius government, cope with the disaster. When we arrived, because naturally much of the effort had been focused on the mitigation and cleanup following the immediate aftermath of the oil spill, very little had been initiated in terms of the monitoring and impact assessment, and particularly coordinating the efforts of all of those that were contributing to that part of the response. And that's really where our skills came in. It was the area that we had the most to offer, really, given our experience from previous premium guidance and the development of that programme. So just back to the type of substance involved. So again, this was mainly the fuel oil from the vessel that was causing the pollution problem. And it was a new generation fuel oil. It's known as very low sulfur fuel oil. And again, because it's less traditionally used, it's quite a new product, it's quite a new fuel type. Relatively little was known about how it would interact with the environment. So this had implications in terms of being able to track its movement within the environment, follow, from the release site, from the wreck itself, into the shoreline areas. The thing that we tend to do in order to establish, where the main areas of impact may be, or the main areas of concern from that sort of pollution perspective, is we tend to use a modelling approach. And that modelling approach, the effectiveness of that, really does rely on knowing the properties of the pollutant and how that would interact with the sea state, the weather conditions, and it's all of that information that we really need in terms of fate and transport modelling, so that we can come up with an accurate prediction or forecast of where the oil will travel and where it will ultimately end up. So one part of our support offer was to do some preliminary analyses on the samples of the fuel oil that we were able to acquire, and that's part of what we did. But then the main area that we were able to support on was really around the integration of the environmental monitoring programme. There were three key things that were needed from this sort of more integrated approach to monitoring. The Mauritius government really did need to be able to carry out a scientifically robust assessment of the environmental impact that had been caused by the oil spill, and also, have in place a monitoring programme that effectively tracked recovery, post-spill and post-cleanup of the local ecosystem in the sort of short, medium and much longer term post-impact. And we also were able to help provide a framework for that sort of more ongoing, longer term monitoring, make sure that everybody's efforts were used as optimally as possible, drawing on the skill sets of multiple contributors. And again, these were the responsible authorities, the NGOs in country, civil society were very much involved as well. And then, of course, the international support community, but perhaps less so in relation to the international teams, in that they wouldn't be there for the long term. So anything that we were able to put in place in that context really needed to be achieved by the organisations that would remain in country and would be carrying out that longer term monitoring programme. So just a brief overview of some of the principles for monitoring and assessment, and this is really taken from those premium guidelines that I mentioned right at the beginning. So the first stage in the process is to establish what the footprint of impact might be, and that's what really draws on those modelling outputs that I referred to earlier. It's really by understanding how the pollutant travels within the environment, in this case, within the nearshore waters and along the coastal areas. So how does it travel? Where is it most likely to end up? What are going to be the main areas that we're concerned about, so that we can do some risk-based monitoring around those areas of highest concern. In order to, again, assess what the potential impacts might be, we need to have a good understanding of the presence and the distribution of those receptors. And when we say receptors, it's basically anything that the oil might interact with and have an impact on. So these can be things like conservation habitats, essential fish habitats, for example, but also commercial fisheries or aquaculture facilities, and also recreational amenities or leisure amenities for the public. Once we've established those two elements, so the footprint of impact and the distribution of some of those receptors that we might be interested in understanding the impact on, we would cross-reference those two bits of information so that we can apply this more risk-based approach to monitoring. We're not going to be able to have the time or the budget to monitor everything everywhere. So it's really important to, very quickly establish where we're going to mainly focus our effort. And those areas are the areas of most concern. So within those areas, what is it that we might actually be interested in monitoring? So for the incident in Mauritius, there were three main areas, and these are quite common things that we would consider for monitoring, regardless of the geographical setting. So the three things that we were really focused in on were the ecology and ecosystem. What were the main habitats and species that we may be concerned about in terms of the impact. In Mauritius, given the sort of tropical setting, we were looking at things like the coral reefs that were adjacent to the wreck site, some of the mangrove habitats along the shoreline. There were also seagrass beds and other ecosystem components, including species that we might have a concern about. There's also the wider environmental quality. So how is the contaminant, the oil in this case, persisting in the environment perhaps? Is it quite quickly broken down, and are we seeing very low concentrations over time, or are there areas where the contaminant really does persist and perhaps will do for a long time? And then also looking at some of the elements around fisheries and food safety. If we've got some important commercial fishery resources or aquaculture facilities, fish farms in the area that we've predicted as the footprint of the impact, then we'd also be interested in looking at contaminant levels within those products in relation to safe levels. And when we say safe levels, it's really around human consumption. So as I mentioned, within the sort of the three monitoring elements, we really sort of did drill down and make it quite tailored to the Mauritius situation. So there were some, in relation to the footprint of impact, there were areas of coral reef, there were areas of mangrove habitat, and there were also fisheries resources within that predicted area of footprints, that predicted footprint of impact. So those were the things that we really focused in on in terms of monitoring effort for Mauritius. There were also a number of impacts that weren't related to the oil contamination that were of concern from a conservation perspective. So the actual wreck itself, when it became grounded, it was actually resting on quite a significant area of coral reef. So naturally, that was a concern in terms of physical impacts. You know, how had the wreck itself impacted the health and, well, the existence actually of some of those coral reefs and coral species in the immediate wreck site. As mentioned before, there was also the oil contamination itself. We can see there one of the maps that were produced as part of the cleanup operation. So it's to direct the cleanup to those areas where we'd seen the greatest oiling. And that actually did match up with the maps that we'd produced from the modelling approaches, which were the predicted areas of shorefall of the oil. So that was validated by the observations of the cleanup teams. But there was also, again, due to the location and the situation of the wreck site itself, there were also concerns about a sediment plume that was being generated and turbidity. You can see there from this overflight image, there's quite a significant and visible area of sediment plume that's moving away from the wreck in the sort of prevailing tidal direction. This again was a concern because the area that was directly within that sediment plume was an area of coral reef, and of course the corals are reliant on you know, being having access to light in order to function properly. And also there's a potential of smothering of the coral reefs on the seabed from that sediment plume as well, which could, you know, also have an impact. So what did we find from our preliminary monitoring efforts? Well, in relation to the contaminant, the oil spill, there was little evidence of oil contamination in those subtidal, subsurface marine habitats. And you'd expect that with oil in the oil floats. So, you know, it tends to be more of an issue at the surface or where it makes shorefall along the coastal areas. So we weren't seeing very much at all in relation to an effect of oil on the coral reefs and also the seagrass habitats that were subtidal. But there were some mangrove areas in the coastal regions that were really subject to quite heavy oiling. Because of the way that mangroves develop and form, they tend to be in those areas that act as a sink, and they're fairly low energy environments. So naturally you would expect any oil to arrive in those environments to be quite persistent and also very difficult to clean up, given that mangrove habitats are quite inaccessible, but also, you know, we wouldn't want to be using any particularly aggressive cleanup techniques, which might actually have a greater impact on the stability and the integrity of the mangrove area. You know, you could end up perhaps doing more damage that way than what the oil might cause itself. In terms of environmental quality, so there was actually quite a lot of baseline information, so pre-spill information in relation to the contaminant, the oil levels in the coastal sediments. These had been monitored prior to the incident occurring, partly through some dedicated monitoring once we realised that the wreck was likely to create a pollution event, but also as part of some of the more routine monitoring that had been carried out by the responsible authorities and also the NGOs. And we were also able to focus in on some of the fisheries resources as well that we were interested in understanding any impacts on. We were able to work with the responsible authority in Mauritius, which was the Albion Fisheries Research Centre, to establish a monitoring programme for some of the fish species that had perhaps been impacted or contaminated as part of the oil spill. So this is just some of the preliminary results that were acquired from that focused fisheries monitoring programme. So just looking forward, in terms of, you know, what's happening now. This all occurred in 2020. We did maintain contact and maintain support for the Mauritius authorities following our return to the UK, working with some of the laboratories and also some of the responsible authorities to continue the knowledge sharing, and also, you know, explore opportunities to continue with that knowledge sharing and you know, with the focus on best practice. Of course, these sorts of pollution incidents are very unwelcome. Nobody wants to experience an oil spill of that significance. But at the same time, it sometimes is an event like this that acts as a bit of a catalyst to, you know, identifying any gaps in knowledge that can perhaps then be filled via some of the more ongoing and routine monitoring programs, which continue beyond the, you know, the effort that's specifically focused on the incident itself. And that's kind of what we're doing now, really, keeping in touch on a more informal basis with some of our counterparts, our technical specialists and scientists in Mauritius, so that we can really continue that best practice knowledge sharing and hopefully prepare for any future incidents, similar incidents, which of course we hope don't occur. It really is a case of, you know, plan for the worst but hope for the best. So thank you for listening. If obviously I can't take any questions at the moment because this is pre-recorded, but if anybody does have any questions, please by all means get in touch. My contact details are there, and also if anybody's interested in having a look at those premium guidelines that I mentioned, there was a link in the previous slide that'll take you to the PDF version online, and you can download that for a better look. Thanks for listening and enjoy the rest of the conference.
Thank you very much, Sue Ware, who can't be here, but we really do appreciate that and that look into emergency responses. So I'm going to hand over to Charlotte, and if there are any questions, please pop them in the chat. I think we are going to move to the Q&A session now because unfortunately I do not see our third speaker in the list of people who have attended. But maybe we'll get him to record it and I can send it to you all anyway, because it will be interesting to hear about the pollution issues in Senegal, to really get an overview of the various different countries. So yeah, I'm going to move over to the Q&A session. And if we can, if you had any questions for the three speakers, if they are still here, if you had any questions for them as well, you can also pop them in. But we're probably going to finish a bit earlier. So I think let's aim to finish it at 1:30, which will wrap up and head for an early lunch or yeah.
There aren't any questions in the chat. Anyone have any questions for Patricia? Hands up, please. Right, so no questions at all. Holly, do you have anything?
I think it was, is Andy still on? He said something.
Yes, Andy said, thanks, Patricia. Lots of thought-provoking material. I know that for mixtures, the assumption has often been that additions rather than synergy is the most likely. These experimental data are critical for ensuring that we continue to consider both possibilities when looking at mixture solutions.
I do have a question maybe for Patricia, who had mentioned, this isn't about ecotox question because that's not my background, but it was a question that I think might relate to all of the different topics that we spoke about today. But we do speak about sustainable financing and data gaps and financing these kind of monitoring programs. And what we're also seeing from, for example, what Andrew was saying about certain sources is a lot of, not always, but a lot of times if we're collecting one sample, how can it be used for multiple pollutants? If we're trying to intervene or understand a source, are we looking at our pollutants holistically? I come from a very non-holistic point of view where I think sometimes as microplastic experts, we do focus specifically on microplastics, and now we're starting to draw in on the metals, but most of that is still in line with association with microplastics. So I don't know if any of the speakers have any anything to add on that point. Patricia, I think you were muted and unmuted.
Right. Okay. Thank you so much for that comment and the question. Well, I actually think that using ecotox bioassays is a really holistic approach because, like, for example, in our study on the effects of the e-waste pollutants, although we were able to analyze for some metals, we know that those are not all the pollutants that are found in the contaminated soil. But however, when we exposed our earthworms, we know that whatever responses we are measuring, we are assessing in the earthworms is the integrated response of all the pollutants present in the contaminated soil, whether or not we're able to identify or quantify them. So I think using bioassays is quite a good approach to complement other approaches, because of course using just bioassays, you may never know what is actually causing the toxic effects. So I think bioassays have a way to, or have something to add to our assessment. And my choice of bioassays are really simple, cost-effective, not necessarily those that may need a lot of funding. Hopefully, we are expecting that maybe in the developing countries, some of these could be adopted to encourage environmental monitoring. I hope that helps.
That does, thank you. I don't know if any of the speakers before wanted to comment on that idea of one sample, multiple outputs and things like that.
Hi, I'd just like to sort of touch on the technologies that could be implemented in regards to pollution, but from a microbial sort of viewpoint. So like the use of functional metagenomics to look at gene expression in specific environments. So like for example, environments that are impacted by heavy metals and other pollutants may be a useful tool to really assess the sort of microbial ecology of these environments. And also you mentioned about sort of ecotoxicological tools that are used. I just want to know a little bit more about pollution-induced community tolerance tests that are to look at basically the microbial communities within these different water sources. So looking at what concentrations of pollutants sort of favour the growth of particular microbes within that environment. And I think this is quite important in regards to looking at some of the natural processes. Like, for example, if the particular pollutants, whether standalone or synergized with others, are sort of having impacts on maybe microbes such as cyanobacteria that are involved in the sort of carbon cycle, and how that has an impact on that sort of cycle and overall impact on sort of climate change, I guess.
I think that might have been a question for Patricia, Andrew?
Yes.
Yes. Patricia, I don't know if you heard that question.
I didn't quite get the question. Please, can you like rephrase it? Not quite sure exactly.
Yes, so I just wanted to know whether in regards to ecotoxicological tools that are used, albeit this is in regards to assessing the microbial communities of some of these environments. I just wanted to know if things such as like pollution-induced community tolerance is used within these environments to assess the concentrations that may be favoring the growth of other microbes over others, and whether there's been any sort of relating it back to sort of some natural processes, such as favoring whether it has a detrimental effect or maybe favors the growth of microbes such as cyanobacteria, pleurococcus, for example, which are involved in the carbon cycle.
Right. I am not aware of tests using those specific microorganisms you've mentioned, but if we remember, Andy mentioned about the microbial assay for risk assessment. I think that is a potential ecotox test, which can be very, very useful in this kind of investigation, because with that test, it is possible to replace the microorganisms that are currently in the test with any microorganisms of our choice. But the whole idea of that test is just unfortunate that I wasn't able to present it given the short notice. But in that, the principle of that test is based on using several microorganisms in the same test system, and exposing whatever environmental media or test substance, and it's an 18 hours test which assesses the effects on the growth of those various microorganisms, which are actually selected based on the diversity of their responses. So I think they could be replaced with any microorganisms of interest, and the test system will be able to assess the diverse responses. For now, it measures growth, but I think at the end, if we want to understand more on maybe the mechanisms or the antimicrobial resistance aspects, I think after that, I am not very familiar with the genotoxic test, but I think that could be combined with the genotoxic test to understand the mechanisms maybe that could explain the various sensitivities of the various microorganisms. So I think that is something that could potentially be done, and I think I'm quite interested in maybe trying to do something along those lines.
Okay, no, thank you. Thank you.
You're on mute, Charlotte.
Yeah, I know. Thanks for your question, Angie. I can see that Andy has posted in the chat, so I'll just quickly read that out, and then I can see we've also got one hand raised as well, so we'll come to that second. So Andy has said, it's possible to collect samples for multiple analysis at the same time, but often you need a different mindset to accommodate the needs of different samples. It's not easy to ensure that one sample or set of sampling scientists will apply the right approach as they move between samples. Microplastics obviously need low plastic, and microbiology needs a level of sterility. You can't assume that the people collecting your samples understand why you use certain approaches or what you are trying to avoid, unless you take the time to really explain before you go on survey, which I think is a very valid point. So move to raised hands. I can see that Lavnan, you've got your hand raised. Would you like to ask your question?
Hello.
Hello.
Can you hear me?
Yes, yeah.
Yes, nice presentation, man. I have one question. Means here I am from India. I'm working mainly on freshwater environmental on microplastic pollution. Here nowadays most of the people working, right? So we have limited facility about micro Raman or micro FTIR. Instead of that, we can use any other techniques like like hot needle test, so that they will confirm with that it's microplastic. They can go with that for polymer composition. We don't have any. We are limited facility here. Most of the people want to use micro FTR, micro Raman, but which cannot accessible. So what could be the another option for them to actually in not needle test they can report, right? Can you give your suggestions on that?
Was your question aimed at someone in particular, or just a question?
Actually, most of the people working right now, microplastic, they want to confirm the polymer type, what exactly, but a limited facility available, here in India. So what will be the alternative, for suppose if you see the hot needle test, so that they can see with it is microplastic, so they can go with that. Anyway, there is a drawback. We cannot confirm the polymer composition, but they can confirm it is plastic. They can say, right? So what could be your suggestion, means how we can go for the
I think we can actually pose that question to all the attendees if you want to answer that question in the chat, because a lot of you are working on microplastics, and I think Anne is using a specific Nile red technique, but then using Raman. I don't know, Anne, if you want to elaborate. We didn't have a specific microplastic talk today, but you've really hit the nail on the head with this idea of harmonized approaches and difficulty with and understanding the polymer identification. So I don't know, Anne, your topic really looks at harmonized approaches across the globe and things.
Yeah, thank you. Hello, it's Anne Robertson here. Yes, we are, I mean, certainly in our first package, if you like, for the global sampling, the collection of the microplastics, we have a standardized sampling procedure, and it's really, critically important, as we all know, for microplastics, so that we can at least understand the contamination issues and so on. As far as actually the analysis of the microplastics goes, yes, we're using an initial Nile red approach, staining with Nile red, but as you rightly say, it doesn't tell us what the composition necessarily of the plastics is, and for that we are going to be using Raman, FTIR, etc., etc. And I mean, this may be my specific ignorance, but I don't know what other approaches there are apart from Raman and FTIR and so on for actually looking at polymer composition. So maybe somebody else might know more. But as I say, we are very much doing a standardized as a protocol for microplastic collection. And certainly with Nile red, you do get some good results in the sense that you can be almost sure that it is microplastics you're looking at. But for the composition, I'm not sure what else you could do. Does anyone else want to chip in?
I think I chip in on that.
Hello?
Sorry. Does
Hello?
someone else? Yes.
Hello, Gideon speaking.
Hello, Gideon. We can
Okay,
hear you.
I just wanted to chip in something. Hello, can you hear me? Can you hear me?
Yes, we can, Gideon.
Yes.
Oh, okay,
we can
okay,
hear
cool. So concerning the question asked about microplastics identification, so I think the question is 2 in 1. If you are talking in terms of how do I know whether this thing is plastic or not, that's from one angle. The other one is how do I know the polymer material that this plastic is made of? So in the first instance, if you want to know whether that thing is plastics or not, of course, you know, by the general definition of microplastics as plastic particles with sizes of 5 mm and below. So there are some plastics in the range of, say, 0.5 mm to 4.99 mm that you can see visibly, physically, and you know those ones are big enough to be seen, and you can see definitely this is a plastic material. But then when you have other particles collected on your filter and you are not sure maybe that's a plastic or not, I think that's one of the essence of using a microscopic technique. So there's this publication, I've forgotten the name of the author, that shows that when you view a particle, you will, if it's not a plastic material, you will see some cell structures. If it's a plant residue, for instance, you will be able to see some plant cells, images that looks like